Hi everyone. This is the second of 3 cardiovascular phys recordings. After these recordings are done, that's the end of the material for your third test. So last time what we did was we went through just sort of the overall sort of physiological purpose of the cardiovascular system and the relationship between pressure, flow, and resistance. And then we talked about autoregulation, and autoregulation is really the way in which a tissue or a capillary bed or a region of the body can increase or decrease blood flow to itself via the release of paracrine agents or paracrine factors that control local vasoconstriction and vasodilation. That's local. It's not really trying to make pressure changes. It's trying to make flow changes due to resistance changes. And really the two reasons that I explained to you guys that it would do that is because the metabolic indicators from the tissue say I need more blood, in that I don't have enough oxygen, or I have too much carbon dioxide, or my pH is too low, or H+ is too high. So that's metabolic control. Any of those things will make the tissue release paracrine agents that can cause local vasodilation. And the other one was sort of a protective mechanism. It's called myogenic control, in which the tissue, if a lot of blood flow is coming to it, the arterioles can constrict to protect the capillary beds. Okay, and then we talked about a few places in the body that have kind of extremes of blood flow. All right, so now what we are going to do is we are going to get into cardiac muscle tissue and the mechanism of action potentials and contraction in 2 different types of cardiac muscle cells. But before we get to that, let's just talk about a little bit of general stuff about cardiac muscle tissue and cells. And so first off, cardiac muscle cells, a few of them actually have the ability to release a hormone. And I know you guys wanted to learn another hormone because you didn't learn enough. This hormone is called ANP or ANF. Oops, not the one I wanted. And ANP or ANF stands for atrial, natriuretic factor, or peptide, or protein. So atrial implies where it's coming from. Natriuretic is kind of telling you the target. And then, of course, peptide or protein tells you the chemical class. So I want to just throw it out to you and see if you can predict what this one would do. So this is a hormone that is released from the atria of the heart in response to prolonged atrial wall stretching. And the natriuretic part kind of tells you what it will, where it will go. It doesn't tell you it implies it. Natriuretic, so the target is actually the kidney. And what it's going to do is it's a little negative feedback inhibition mechanism in which the heart is encountering prolonged lots and lots of blood in the heart. And it's going to target the kidney. And do you think that it should target the kidney to increase or decrease blood pressure if it is released as a response to prolonged atrial wall stretching, just based on what you know before? And of course, natriuretic implies sodium in urine. So what it's really going to do, guys, by the way, is it is going to cause you to, oops, I did that one wrong, to help decrease blood pressure. Flip that one around, Melanie. Hold on just a second, I'll go find a down arrow and stick it in there. I was trying to do that beforehand, so I had less farting around to do in the middle. And of course I messed it up because that's how I roll. So it actually targets the kidneys to decrease blood pressure. And again, it is a negative feedback inhibition mechanism. And really what it does is actually causes the kidneys to excrete more sodium than they otherwise would have. and that is going to be a mechanism of decreasing the blood pressure. So you detect too much blood in the walls of the atria, you release ANP, goes to the kidneys, tells the kidneys to pee out more sodium and therefore more water, and your blood pressure goes down. Okay, so next thing, cardiac muscle cells, what about their metabolism? Meaning, what do they need in order to do cellular respiration? They're actually not very picky about nutrients. So they can burn glucose, other sugars, they can burn fatty acids, they can burn lactic acid, they can use almost any of these nutrients for energy. So getting nutrients to cardiac muscle is not generally a problem, but they have almost zero capacity for anaerobic cellular respiration. So they are, because of that, they are intolerant of ischemia. And ischemia is just this fancy word that means reduced blood flow. It's the opposite of hyperemia. So whereas they're not at all picky about nutrients, they are super duper picky about oxygen. And we of course know that cardiac muscle cells are, for all practical purposes, amitotic. So if they do not have oxygen for a decent period of time, not only will they die, but you don't get them back. Okay, and then now we're going to get into this two specific categories of cardiac muscle tissue, cardiac muscle cells. There are two different kinds of cardiac muscle cells. There are what we call pacemaker cells. Your textbook tends to refer to them often as autorhythmic cells, as in they make their own rhythm. And of course, these cells are clustered in nodes, SA node and AV node. And then there are also chains of them in the AV bundle and the left and right bundle branches. And I'll show you an animation in just a second, but what these do is during embryonic development, they actually lost their contractile ability. Instead, they became highly specialized not to contract, but to depolarize. And they depolarize rhythmically. And after they depolarize, they actually send the ions that they use for depolarization through gap junctions to their neighboring cells. And if their neighbor is another pacemaker cell, then they just transmit it through the pacemaker cells. But when they get to the Purkinje fibers, for instance, they will communicate to the contractile cells. So let me show you guys an animation of that just to get you started on it. So anyway, they are cardiac muscle cells, but they lost their ability embryonically to contract, and they don't even have actin and myosin in them. So let me show you how those work. All right, so you learned this in anatomy, but I'm just going to give you a little visual refresher as well.
Action potentials originate in the sinoatrial node and travel across the wall of the atrium from the sinoatrial node to the atrioventricular node. Action potentials pass slowly through the atrioventricular node to give the atria time to contract. They then pass rapidly along the atrioventricular bundle, which extends from the atrioventricular node through the fibrous skeleton into the interventricular septum. The atrioventricular bundle divides into right and left bundle branches, and action potentials descend rapidly to the apex of each ventricle along the bundle branches. Action potentials are carried by the Purkinje fibers from the bundle branches to the ventricular walls. The rapid conduction from the atrioventricular bundle to the ends of the Purkinje fibers allows the ventricular muscle cells to contract in unison, providing a strong contraction.
Okay, so that was just a review of kind of stuff you learned in anatomy. I just wanted you to remember where those pacemaker cells are. So in addition, I also want to point you to this. I changed these links so that they're more updated based on the access that you have to your textbook. I hope the links will work for you. If they don't, then you have to go through mastering and get into interactive physiology, but let me just show you kind of what they do. This is the electrical activity of the heart, and when it's an interactive activity, it means it's not just an animation. You can do stuff with it. Let's see if this one will work. I think this one's really, really good. It goes through the intrinsic conduction system, and then what we're about to do right now is an activity on the pacemaker cells. So it has you drag things, it has you turn things up, it has you turn things down. It's a really good way to reinforce the information. And I think this one's worth it. So the ones that I thought were really worth your time, I linked in your notes. Again, don't email me if it's not linked. Just go into Mastering and find one that is IP2, electrical activity of the heart. If you truly can't find it, ask somebody else. You can post it on discussions, and I'll try to make you a video of how to get there. Okay, so I want to talk about the process of generating an action potential in these pacemaker cells. We are not going to be in the contractile cells yet, so no real actin, no real myosin, nothing like that to consider. All we need to do is we need to spontaneously generate an action potential and send it through gap junctions to our neighbors. And that's what's going to happen at the SA node and the AV node and all of the bundle and bundle branches. So first thing is the pacemaker cells, or autorhythmic cells, or sometimes called nodal cells. What they have is a resting membrane potential that really never stays at the same point. What it does is it continuously, a variable resting membrane potential, it averages out about minus 60 millivolts, but it's constantly doing this. Variable resting membrane potential of about minus 60 millivolts. But what happens is if you have a dependable sodium electrochemical gradient, and you work really hard on having a dependable sodium electrochemical gradient, then what's going to happen is you are going to have these sodium channels that are always in the pacemaker cells. And if you have a dependable number of these sodium channels, they're weird channels, but and if you have a dependable sodium electrochemical gradient, then what you're going to have is a dependable rate of depolarization, and then you'll hit threshold, and then you'll have an action potential, then you'll repolarize. But again, you've got these channels, so you're going to just depolarize again and again. Now, these channels are in addition to the leak channels that establish the resting membrane potential. So let me tell you a little bit about these. Okay, so what sodium is going to do is it's going to leak in slowly but dependably through, they call them funny channels because they're kind of like leak channels, but there are circumstances in which they might close. So they're not really leak and they're not really gated, so they are literally called funny channels. So what's going to happen as the sodium leaks in through the funny channels, which are right here, the potassium channels are going to be closed at this point. And then what happens is when you leak to threshold, and threshold is, people can't quite agree, between minus 40 and minus 50 in cardiac muscle cells. This one shows minus 50, but most of the literature says minus 40, but sorry, I won't ask you either one. Just want you to know it's the right direction, okay? As you begin to depolarize and you hit threshold and cardiac muscle cells, what happens is you're going to open a new kind of channel. And this is a channel that you haven't met before either. This channel is called a fast voltage gated calcium channel, because you'll meet a slow one later, okay? Fast voltage gated calcium channel. This is going to open the fast voltage gated calcium channel, and then this upsweep that you see right here is caused by the fast voltage gated calcium channels. Calcium is going to rush down calcium's electrochemical gradient, which will move it into the pacemaker cell, further depolarizing the pacemaker cell. Contraction does not follow in these cells, and that's really important. What does happen, however, is that before I'm talking about repolarization, let's just acknowledge that's what's going to happen, is after I have flooded myself with sodium and calcium, I'm going to pass it on to my neighbors through gap junctions, because of course cardiac muscle cells are connected with gap junctions. So contraction doesn't follow. They are going to pass it through gap junctions to their neighbors, and if their neighbors are contractile muscle cells, we will talk about how that causes them to contract. But before we get to what the contractile cells do, let's go ahead and acknowledge that you're going to have to repolarize this cell if you're ever going to use it again. So what's going to happen is this, again, was the sodium funny channels, okay, and they're kind of leak channels. This is, of course, threshold, minus 40 to minus 50. And then this is caused by opening voltage-gated calcium channels. They are the fast voltage-gated calcium channels. And then how do you imagine we would get back down to this variable resting membrane potential? We're going to have to close our calcium channels, right? We are going to have to open, what kind of channels do you think could get me right back down here? These will be voltage-gated potassium channels, because potassium always helps you get back to resting membrane potential from a depolarized state. These are going to be voltage-gated calcium channels. And then, of course, we're going to have to pump, but we're going to have to pump sodium, potassium, and calcium. So what you are going to have is one more pump that you hadn't met before. You are going to use the sodium and potassium pump to get most of the way back down here, but you still have to get rid of whatever calcium didn't leak through the gap junctions. So you're going to have to pump calcium back out as well. So sodium, potassium pumps, and calcium pumps. So now let's just make sure that you can label stuff on this in your notes. Okay, so label on figure 1 the sodium channel. So let's find our sodium channel. And of course we call it the funny sodium channel. So what's going on from here to here? This, damn it, why does it keep doing that? This guy right here is the sodium channel, right? That's an open, it's generally going to be open, but that's a sodium funny channel. And then I asked you to label the calcium channel, and the calcium channel is right here, and it is a fast voltage gated calcium channel. And then what happens here is you close calcium, and then this repolarization sweep is primarily the potassium channel. So one more time, sodium channel, funny channel, fast voltage-gated calcium channel, and then voltage-gated potassium channel, and then do the whole blasted thing over and over and over and over again. So this, you've heard before that the SA node depolarizes to threshold about 100 times per minute, right? This is its inherent rate of depolarization based primarily on the number of sodium funny channels that you have and your electrochemical gradient for sodium. So that occurs about 100 times per minute. But let's go back to the last stuff that we did, and we can say, okay, but I know that the autonomic nervous system can increase or decrease that rate of depolarization. And I also know that in a healthy person, your heart rate is not 100 beats per minute at rest. So what does the nervous system have to do with this story if it works on its own? And it does work on its own. But what does the nervous system do? What the nervous system does is, like I mentioned before, the metaphor that I like to use is that the SA node starts the car, but the nervous system can either put the gas on with the sympathetic nervous system or the brakes on with the parasympathetic nervous system. So let's see what that actually does. So let's first look just at the hookup real quick. Okay. So what we see, again, reinforcing the last stuff that we did, is that the medulla oblongata is really going to be in charge of this whole story. Okay. And the medulla oblongata, via the vagus nerve, is going to attach the parasympathetic nervous system to the heart via the SA node and the AV node. If this goes to the heart and releases acetylcholine onto the SA node and the AV node, what it will do is slow the rate of depolarization. Did I pull that picture up? Oh, shoot. There's a picture I want to show you. Always think I'm so ready, and then I'm just not. So hold on just a second. Wait right there. 13. I'll show you what it does. Okay, this is it. Okay, so this looks so busy, but it's just fine. Okay, so what's going to happen is the parasympathetic nerve will take this like rhythmic depolarization, and it'll keep it from hitting threshold as quickly and therefore slow the heart rate. The sympathetic nervous system would make it hit threshold more quickly. and then it would have a faster heart rate. So I want to show you this figure real quick. Okay, let's get rid of this. So I'm going to use one that's a different color. All right, so what we have is this one right here is what the parasympathetic, what the SA node does without nervous system stimulation. It hits an action potential, it repolarizes, it hits an action potential, it repolarizes. That one's in gray. That's when you don't apply any nervous system activation or inhibition to it. But what happens is if I apply the parasympathetic nervous system to it, it's this blue one. Notice that with the blue one, it takes you longer to get to an action potential, and then I don't even see the next action potential in this one. And the way that it does that, by the way, is by releasing neurotransmitters, acetylcholine, at the cardiac muscle cells of the SA node and the AV node. And whereas they were on their way to depolarizing to threshold, it slightly hyperpolarizes them. It makes them go the other direction, which means it's going to be longer before it hits threshold. Okay, and the sympathetic nervous system, however, if you were on your way to threshold and then you release a neurotransmitter, norepinephrine, that makes you hit threshold faster, then notice that you hit threshold and get an action potential much faster. So if this one was at rest, depolarizing about 100 times per minute, when I stimulate it with the parasympathetic nervous system, it can slow down to 80, 65, whatever. But if I hit it with the sympathetic nervous system, it can go up to 120, 130, 140. Okay, that's how that actually works. And so that's just looking at all three of them at the same time. So let's just make sure that we understand the part in your notes. Okay, so the rate of excitation and therefore contraction in the pacemaker cells is controlled partially by the autonomic nervous system. Go back to our autonomic picture here. Autonomic nervous system. And what the autonomic nervous system does is parasympathetic hooks up to the SA and the AV node. Sympathetic, medulla oblongata, controls it, but it controls it via going down to, again, the thoracic region of the spinal cord, and then out through the ganglia, right? And then to the SA node and the AV node. And it's going to release norepinephrine, and that's going to increase the heart rate. Now we will see this too. This is going to be important to us a little bit later because the sympathetic, not the parasympathetic, but the sympathetic doesn't just go to the nodes, it also goes to the wall of the ventricles. That's going to be important for us just a little bit later. Okay, so there is a good interactive physiology animation. I'm not going to play this, I'm just going to show you what it looks like, that goes over the process of generating an action potential in these cells, show you what it looks like. And it goes through and shows you the ion channels at the same time. And this one specifically focuses on the autorhythmic or pacemaker cells, and then it leads into the next step we're about to talk about. Okay, so all this fancy stuff that we've talked about, and we haven't even contracted yet. Okay, all we did was depolarize and then send it through gap junctions. So now, of course, the next thing that we're going to talk about is what about the contractile cells? What about the cells that actually do the contraction in cardiac muscle cells? So we need to talk about how we get those to an action potential and then how the action potential causes a contraction. So first thing is the depolarizing stimulus that we just received. So let's look at this figure. This one should look kind of familiar to you because it's drawn really similarly to the one that we looked at with contraction in skeletal muscle, and that was on purpose that they did this. So I want to teach you how it works and then show you what the action potential looks like. So please note that one of the major similarities here is calcium, calcium binding to troponin, moving tropomyosin out of the way, and then you get cross-bridge cycling as long as you have ATP. So that's really similar. What is immediately different is that, of course, I don't have a motor end plate that is actually responding to neurotransmitters, and I don't actually need one because, of course, these cells get flooded with ions not from neurotransmitter binding to them, but from their neighbors, right? So they are going to get the ions that will cause them to have a depolarization and an action potential. They're going to get that from their neighbors. And let's say their neighbor was a pacemaker cell. Okay, so the depolarizing stimulus is going to be a bunch of sodium that came in to the neighbor through the sodium funny channels, and then a bunch of calcium that came in through those fast voltage gated calcium channels. Here I was just talking about pacemaker cells, not contractile cells. So I've got a whole bunch of sodium and a whole bunch of calcium, and what's going to happen is that is going to go through gap junctions into the contractile cell, and it's going to almost immediately make it hit an action potential as soon as it goes in there. So their graph of their action potential in a contractile cell looks very, very different than it does in other cells. So first I'll show it to you here, and then we'll look at it in more detail. These guys are generally sitting around at about minus 90 millivolts, the contractile cells, and then they get flooded with sodium and calcium, and they have a really quick depolarization. Okay, so let's talk about the ions that are involved with those. Okay, so we'll come back to causing contractions. So what's going to happen here is it's going to get flooded with, sorry, I lost something. Oh, there it is. Okay. So the depolarizing stimulus, as soon as it comes in, will cause you to almost immediately hit an action potential. And that occurs when we call these fast voltage gated sodium channels open. That's these guys right here. The fast voltage gated sodium channels open, and you get almost an immediate action potential. And you get an action potential. And then what's going to happen, let's just look at this and we'll go back to see what happens inside the cell with the contraction. The action potential is going to move along the plasma membrane, but then it'll also go down the T-Tubules in just a second. But the other thing that makes this kind of strange is that action potential is fast voltage gated sodium channels, and then it starts to repolarize just a little bit. I won't have you memorize how it starts to repolarize, but then what happens is that actually causes the opening of a second kind of channel that actually keeps the depolarization going for longer. And this channel is going to be a slow voltage gated calcium channel. And the slow voltage gated calcium channel will make sure that this cardiac contractile cell has a long, strong action potential. No wimpy little action potentials, long, strong action potential followed by a long, strong contraction. So the original stimulus also causes the opening of slow voltage gated calcium channels, thus allowing calcium from the extracellular fluid in addition to the calcium that came in from the gap junctions, and this prolongs the action potential. Okay, now all that calcium can, for instance, bind to troponin and tropomyosin in addition to the stuff that's actually leaving from the sarcoplasmic reticulum. So let's close all of this first, and then I'll go show what happens inside the cell. So finally, what's going to happen is that our sodium channels are going to start to close. They close all along here. Our calcium channels are also going to need to close. And then I'm going to have to start to repolarize right here with what kind of channels would you use to repolarize? What kind of channels do you always use to repolarize? Those are going to be voltage-gated potassium channels, getting me back to rest. Now, of course, you are going to have to put everything back where you got it. So I am going to have to pump sodium and potassium, right? Okay, but then I'm also going to have two different calcium pumps because I got part of the calcium that I used from the extracellular fluid, and I got part of the calcium that I used from the SR, and I'm going to have to pump it back into both places. So we're going to go through this now here, and then we'll label it in your notes. Okay. All right, so now let's follow along with this that kind of shows you the whole thing. All right, so this is the whole story showing both where the ions are coming from and also how it causes contraction. So you ready? So first, the current meaning the sodium and calcium, comes in through the gap junctions of the contractile cell. That's number 1 right there. And then 2, the action potential gets started if you hit threshold, and it's going to go down the sarcolemma, and it's also going to go down the T-tubules. But as it goes by the voltage-gated calcium channel, it's going to cause that to open. And when that occurs, you get that plateau that allows you to have a longer, stronger action potential and more calcium. Okay. Then that calcium and the action potential is going to keep going down the T-tubules. Calcium is now coming in from the extracellular fluid, but as you go down the T-tubule, that calcium is also going to be caused to be released from the sarcoplasmic reticulum. And wherever calcium came from, whether it was here, here, or here, it's all going to do what you expect it to do. It's going to bind to troponin, move tropomyosin out of the way, then you will start cross-bridge cycling, right? And you'll do cross-bridge cycling for as long as you need to. Then when you are finished with the cross-bridge cycling and the contraction, then what's going to have to happen is you are going to have to put calcium back where you got it, right? And you're going to have to put calcium back into the SR. There's one of your calcium pumps that goes back into the SR right there, okay? Then you're also going to have to pump calcium back out into the extracellular fluid. And I won't hold you responsible for this sodium calcium pump because there's also one that's partnered with sodium. And although this isn't showing you the sodium potassium pumps, those also have to be working. And then of course, when you get rid of all the calcium, you know what happens. Then the tropomyosin slips back into position, right? And then the sarcomere lengthens, and then you're back to your original relaxed sarcomere position. So let us go through the contractile stuff on this one. So let's label on this figure the major channels that are involved. And also you should be able to recognize that this one that kind of looks like a mountain or a bluff, I guess. This one is a contractile cell, whereas this one right here, whoop, whoop, kind of like ocean waves, that one is an autorhythmic or pacemaker cell. So let's make sure that we're confident with labeling these on this figure. All right, so if I needed to, let's remove these. Okay. If I needed to, could I label the fast voltage-gated sodium channel? Yes, I could. This is the fast voltage-gated sodium channel opening. A little bit of a repolarization. I won't ask you how that one occurs. And then I need the slow voltage-gated calcium channel. That's this one that creates this plateau right here. And then this is a voltage-gated potassium channel that causes repolarization. And this is the cardiac contractile cell. That's what those are. Okay, so that is the cells. I would highly recommend you guys going through the interactive physiology activity about the electrical activity of the heart. And if that one still doesn't make sense, you can watch this animation as well. They're all relatively short, just enough to sort of reinforce the material. And then the last thing that I want to talk about before we kind of change gears and start talking big picture blood pressure and blood flow regulation is refractory period. So what we don't want to occur in our cardiac muscle cells is we don't want temporal summation in which we get a weak depolarization that I can add to and add to and add to so that I can actually lead to incomplete or complete tetanic contraction. I don't want my heart to go into tetanus. I want a strong contraction, but I want a strong contraction and then a forced relaxation. So one of the ways that you can make sure that cardiac muscle is not able to do that is that you can force a very long refractory period after each action potential. If you're in a refractory period, you can't get another action potential no matter how much neurotransmitter or how much you flood it with, because the voltage-gated channels aren't reset yet. So the refractory period for cardiac muscle cell is way longer than it is for skeletal muscle. and it's about 250 milliseconds compared to 1 to 2 milliseconds for skeletal muscle. This prevents summation and tetanic contractions. So if you can't add graded potentials because you're still in the refractory period and your voltage gated channels have not reset, then you can't get summation and tetany. Summation and tetany would prevent stop the pumping action of the heart. So unlike skeletal muscle, the heart has really no capacity for summation, and therefore there's no tetanus possible because of the long absolute refractory period. And that is really important because the heart, when it's relaxing, it's actually filling, especially the ventricles. The other thing that's happening when the heart is relaxing is that is the only time that the coronary arteries can deliver blood into the capillaries of the myocardium. Because when the ventricles are contracted, the blood gets pushed out into the aorta and then into the right and left coronary arteries and into some of the other arteries of the heart. But at the same time, it's actually compressing the capillaries themselves. So you can't really get blood into a capillary when the cardiac muscle is contracted. It's kind of like squishing a sponge underwater. The sponge doesn't actually take up the water until you relax. So you have to relax the cardiac muscle in order to let the blood into the capillaries. So the point being, cardiac muscle contraction is just as important as cardiac muscle relaxation is just as important as cardiac muscle contraction. Okay. So now what we are going to do is we are going to change gears and get into a concept called cardiac output. Okay, so let us, before we get into cardiac output, revisit this thing that some of you guys will like and some of you will hate. Doesn't matter if you learn this way, it's fine. It's a decent way to teach even if you don't ever look at it again. Okay, so what we said last time was that blood flow is basically an interaction between pressure and resistance, yes? And that generally speaking, pressure favors blood flow and resistance opposes blood flow. And then what we said last time was that we learned a little bit about resistance, and we said, okay, the primary factors impacting resistance are the vessel diameter, the viscosity of the blood, and the vessel length. So we already did that part. Okay, so what are we going to do today? Well, what we're going to do is figure out that, okay, the pressure that we said was responsible for blood flow can be primarily accounted for by three things. What the heart does. In a minute, I'm going to call that cardiac output. What the blood volume is, that's primarily controlled by the kidneys. And then that pressure can also be augmented or reduced by changing the vessel diameter. Okay, but here what I want to do is concentrate on pressure being generated by the heart, which is probably not confusing to any of you guys at this point. So we're going to call that cardiac output, and let's define cardiac output in a little bit more detail. The cardiac output is really the amount of blood that is ejected from each ventricle per minute, per unit time. Doesn't have to be a minute. And it's generally measured in some unit of volume over some unit of time, usually liters per minute. Okay, so what I'm saying is what leaves the heart is basically going to be measured in volume per unit time. And we're going to call that cardiac output. And cardiac output is generally determined by two factors. The heart rate, and the heart rate is the number of times your ventricles contract per minute, yeah? And then the stroke volume is how much leaves the heart per squirt, okay? So hopefully that makes sense to you. Let's highlight that to make sure that you get it. So the equation is really simple, okay? The equation is cardiac output, CO, equals heart rate, how many times you contract per minute, times stroke volume, and stroke volume is how much blood leaves per contraction. Okay, so here's just an example of how cardiac output can actually work. Okay, so an adult cardiac output example that's within reason is a stroke volume of about 0.7 liters per beat, just to keep it in liters, which is around a quarter cup per squirt. And usually we're concentrating on the left ventricle unless somebody's specifically making you look at pulmonary circulation. So about 0.07 liters per beat. And then a heart rate that is within the normal range is about 72 beats per minute. So if you calculate that out, you end up getting around 5 liters per minute of blood being pumped for your cardiac output, which is pretty damn impressive because that's at rest and you only have about 5 to 6 liters of blood. So your heart is, even at rest, pumping almost your entire blood volume every minute. Okay, so now what I want to start talking about is I want to talk about heart rate and stroke volume. Okay, and so let's get back to our figure here for just a second. So cardiac output is basically heart rate times stroke volume. Okay, we already know quite a bit about how you control heart rate. We know that primarily it is the sympathetic and the parasympathetic nervous system controlling the rate of depolarization in the SA node and the AV node, and we kind of just talked about it. We'll talk about it in more detail in just a little bit. But what you might not know, and takes a minute to understand, so I want to do it first, is what controls stroke volume. So I want you to have a big picture understanding of stroke volume before we get into the weeds and then get right back out of the weeds. So your big picture understanding of stroke volume is every time my ventricles contract, blood leaves my heart. But what is it that controls how much blood leaves my heart every single time the ventricles contract? Not heart rate is a totally separate thing. So per squish, how much leaves and what controls that? So what are the factors that control stroke volume? And that's what we're going to talk about right now. So each of these factors, heart rate and stroke volume, has multiple things that control it. So cardiac output control is really complicated, although if you separate it out into stroke volume and heart rate, it gets a little simpler. So the first thing that we are going to talk about is what controls stroke volume. Okay, so let's open this one up. Okay, all right, so we're going to talk about these, and notice this one's red. That means that's going to decrease stroke volume. These two are green, though they're going to increase stroke volume. This one's probably the easiest one to understand of the three of them, as long as you were actually listening. Okay, so the factors that impact stroke volume. The first one we are going to call afterload. And so what I want you to think about with afterload is that, okay, what's going to happen is that my ventricles are pushing blood into my arteries, yes? Okay, ventricles push blood into the arteries. But an artery can either be, your arteries collectively can either be nice and wide open, or they can be all gunked up and narrow. If they're all gunked up and narrow, or if you have major vasoconstriction going on in your arteries, then the pressure that you're pushing against from the heart is higher. And remember, blood flows from high pressure to low pressure. And so if, let's say, the left ventricle is generating a pressure of 120 millimeters of mercury, but the pressure in your arteries that you're pushing against is 105, there's not a very big delta P. So what it's going to do is, if the pressure in the arteries, which we call afterload, is really high, it decreases the delta P, so less blood is actually going to leave. That's called afterload. So let's look at it in your notes. Afterload is the arterial pressure against which the ventricles must pump. It's the pressure that they're pushing against. It's not the pressure that's generated by the ventricles. It's the pressure that's residual in the arteries. And so what do you guys think? Increased arterial pressure tends to do what to stroke volume? Okay, so think about it. Increased arterial pressure tends to do what to stroke volume? If the pressure is higher here, then what's going to happen to the stroke volume? Okay, So what's going to happen to the stroke volume if the pressure is higher in your arteries is that it drops the delta P and you are not going to have as much flow, right? So what happens here is increased arterial pressure tends to decrease stroke volume, okay? So as the pressure gunks up in your arteries, your stroke volume tends to drop. And this ends up being clinically important because with hypertension, what happens is it drops stroke volume. And if you need to maintain the cardiac output and your stroke volume drops, what are you going to do to your heart rate, guys? So what tends to happen with heart rate with chronic hypertension? Your heart rate tends to go up, right? That's just to maintain this. This cardiac output, because of cardiac output is one of the primary determinants of pressure, which is needed for blood flow, and blood flow serves capillary beds, and that's the function we are all serving. That is the Lord of cardiovascular system, is ensuring adequate blood flow to capillary beds. Okay, so that's afterload. If you increase afterload, you decrease stroke volume. But now we'll get to the one that you will not understand the first time you go through, so be patient with yourself, pause, try to get the little pieces, go back and try to add more pieces to it. It's fine. It took me a while to piece this out to figure out how to teach it, so be nice to yourself. Okay, so the next concept that I want to talk to you guys about is the concept of end diastolic ventricular volume. Okay, this is the volume of blood in the ventricle, so this is called EDV. The volume of blood in the ventricles just before contraction. So before we get into the nitty-gritty of this, let's talk about what should be true. So let's talk about systole and diastole. Systole is a fancy word for contraction, and diastole is a fancy word for relaxation. So question for you. When should the ventricles be fullest? Should they be fullest at the beginning or end of diastole? So, end diastolic ventricular volume. So at the beginning of diastole, when it's relaxed, that is when the blood is just starting to fill into the ventricles. But at the end of diastole, right before systole, is when the ventricles should be fullest. Okay, so let's go back to trying to figure this out. So what is the relationship between the end diastolic ventricular volume, the EDV, and the stroke volume? Well, it's a positive relationship, right? The amount of blood that you have in your ventricles at the end of diastole, right before systole, is directly correlated to the stroke volume, right? So how much blood you have in there is going to be directly correlated to how much squirts out, okay? But one of the things we need to understand is that the ventricles are not just a rigid chamber for blood. They actually can get full and they can get fuller, okay? So that's important to understand. So it has to do not just with the volume of fluid, but also with the volume of the container, how much fluid it can hold. So let's look at the factors that actually impact EDV. First of all, let's see what your prediction is. The volume of blood in the ventricles just before contraction at the end of diastole. What do you think? Increased EDV tends to do what to stroke volume? Increase or decrease stroke volume? What's your prediction? Increased EDV, increased amount at the end of diastole in the ventricles, both the size of the chamber and the amount of fluid that's in it. Increased EDV tends to do what to stroke volume? Let's see. Increased EDV tends to increase stroke volume. So there's a direct correlation between EDV and stroke volume. But now we need to get to the factors that impact EDV. So one of them is called preload. All right. So stick with me for a second. So the ventricular end diastolic pressure. So remember, afterload occurred in the arteries because if you're looking at circulation, can we agree that arteries come after ventricles? Okay, so afterload occurred in the arteries. Preload is going to occur in the ventricles. So the ventricular end diastolic pressure, also called preload, has to do with how full the ventricles are, the amount of tension in the cardiac muscle cells of the ventricles just before ventricular contraction. So as preload increases, stroke volume increases. So get them fuller and you're going to get more in there and therefore more out. Okay, so preload, how full the chamber is. Okay, so preload, how full the chamber is before contraction. And what preload has primarily to do with is how long it had to fill, and then how hard the atria are pushing blood into the ventricles. So let's look at this figure for a second. Okay, so we're going to have preload. That's what we're trying to deal with here. And preload primarily has to do with filling time. If your heart rate's going really, really fast, you don't have time to fill the ventricles before they contract. So if you slow down and give it a little bit more filling time, you'll get the ventricles more full, have a bigger EDV and a bigger stroke volume. And then the atrial pressure has to do with how much blood is coming back to your atria, and then how well your atria are contracting to get blood into the ventricles. Okay, so if you didn't have much blood going back via venous return to the atria, then that's going to drop your atrial pressure, which will drop your preload, okay, which will drop your EDV, okay? Or if your heart rate was going too fast, then that would drop your preload and drop your EDV. So filling time is dependent on heart rate, and as heart rate decreases, not so slow that you're going to get dead, but as it decreases so that you have more filling time, as heart rate decreases, filling time and preload both increase. And then with atrial pressure, what's in your atria primarily has to do with venous return. Venous return kind of has to do with what's going on with your skeletal muscles and your respiratory tract. And then atrial pressure is when the atria contract and push that last 20% of blood into the ventricles. So EDV has to do with preload has to do with filling time and atrial pressure. I have atrial fibrillation. And with atrial fibrillation, what happens is the ventricles are doing just fine, but the atria are spazzing out. I want to take a second and see if you guys can figure out what happens to stroke volume, and then what you do to compensate with someone who's in a bout of atrial fibrillation. So with atrial fibrillation, what's happening, if it hasn't been going on for a super long time, is that the ventricles are contracting just how they should, but the atria are spazzing out. They are not contracting hard, they're just kind of butterflying. It truly feels kind of like butterflies in your chest. So which of these things in the story that we've just been telling do you think that is going to impact? So starting from the outside, atrial fibrillation, can we all agree, is going to drop the atrial pressure? Not so much because there's not blood coming back, but I'm not actually pushing that 20% of blood into the ventricles. If you drop the atrial pressure, you are going to drop the sort of amount of blood that's in the ventricles. Remember what our preload was? The amount of tension in the cardiac muscle cells of the ventricles, how full they are before contraction. And if you drop how full they are before contraction, then you are going to drop the end diastolic ventricular volume, right? And when you drop EDV, you drop stroke volume. And so my stroke volume falls by about 20% really quickly. But I still need to maintain cardiac output because I've still got to serve the god of blood flow, right? So what happens in an atrial fibrillation situation is that after it's going on for just a second, then my heart rate starts to go crazy to compensate. So my heart rate goes from 60-ish beats per minute to up to 115, 120. And we're doing that so that we can maintain the pressure to maintain the flow. Hopefully that gives you a good understanding of how this whole stroke volume and heart rate stuff is related. I think it's a good example. Okay, so now we get to what's called Starlings Law. And Starlings Law has this interesting thing that is related to all of this stuff. So let's talk about Starlings Law. Starlings Law is this length-tension relationship in cardiac muscle cells. And there's actually a good way to review Starling's Law that I'll show you in just a second. Okay. All right. So Starling's Law is going to impact EDV as well. Okay. And it's got some relationship to this other stuff. But basically what Starling's Law is going to say is that the heart actually pumps blood out better when the ventricles are stretched. So stick with me and I can show you how this works. Okay, so Starling's Law, let me get rid of these. Okay, so first off, I'm going to post this for you unless one of you guys posts it first. So, where are you, Starling's Law? This same book that I keep showing you guys, and I know that Donna ordered hers, it's still pretty cheap. I want to show you a couple of things that it has that I'll try to get to post for you today. This is a practice activity over action potentials in cardiac muscle. It's a labeling. This one right here is related to how cardiac muscle causes contraction. And then this one, it's called the Frank-Starling Law because there was a guy named Frank involved in it too. This is exactly what I'm going to show you right now, the Starling Law, and a way to practice it. So Starling's Law says that there is a natural way for the heart to contract harder and cause greater stroke volume when it's more full. Okay, so let's show this one. Okay, so stick with me. This is called Starling's Law. Okay. So basically you've got two different sarcomeres right here. Okay, and these are cardiac muscle sarcomeres. They look just like skeletal muscle sarcomeres for all practical purposes. And notice that what this is showing you is that as the EDV goes up, as the heart gets fuller, then you actually get a greater stroke volume. So this is basically a cardiac muscle cell when it's not very full. Okay, so the EDV isn't very big and the stroke volume isn't very big. But what happens, and this is naturally, and this is not nervous system controlled, it's not paracrine agents, it's not anything. This says that as you get more blood into the atria and therefore into the ventricles, what will happen is you get your cardiac muscle cells, the sarcomeres, to an optimum length so that they have more access to cross-bridge cycling. Can you see that just theoretically between this one and this one you have more access to cross-bridge cycling? So what's the deal is with cardiac muscle cell is that cardiac muscle sarcomeres actually contract better when they are stretched than when they are not stretched. So as venous return changes, as you get more blood into the atria and therefore into the ventricles, the heart automatically adjusts its output to match the inflow, which prevents enlargement of the hearts, because when will you contract best? When you are very full. Cardiac muscle sarcomeres have an optimum length, and they hit that when they're stressed. So basically what's going to happen is all the blood you get in is going to stretch them to their optimum length, and then they are going to contract better. So this increased venous return that goes in, for instance, when you are exercising, what causes venous return to come back? When we're exercising, we of course know what causes the blood to go out because of course, primarily that is an increase in heart rate, and then also we'll see it's an increase in contraction. So I've got a whole bunch of blood going out. Okay, and that directly makes you understand what happens with exercise and arterial flow. But what happens with exercise and what we call venous return? I'm going to define venous return one more time for you. Venous return is blood returning through the veins to the atria of the heart. What causes the blood to return really well, like during exercise, which will cause the heart to be really full, is a couple of things. Venous return is aided by what we call the skeletal muscle pump. So when you are using your muscles, what they do is they push on your veins, right? And since many of your veins, especially in your appendages, have one-way valves in them, what that is going to do is the more you squish them, the more you are going to push blood up the vein, and it can't go backwards because it's a one-way valve. So basically what you're doing is you're sort of moving that blood back up your veins, back into your inferior vena cava, and then back into your atria. Okay, so that's called the skeletal muscle pump. So not only is your arterial flow better when you're exercising, your venous return is also better. And then there's also what we call the respiratory pump. And the respiratory pump is just kind of cool. They didn't have a good picture of it in your textbook, but I think I can explain it to you. So here's the deal with the respiratory pump. What happens is when you breathe in, your diaphragm moves down, right? And then, so when you inhale, your diaphragm moves down. When you exhale, your diaphragm relaxes back up, moves down, relaxes back up. Well, going through the diaphragm, among other things, is your inferior vena cava, which of course is delivering the vast majority of the blood back to your right atrium. Okay, so what happens is the inferior vena cava is going up through the diaphragm, and when I push the diaphragm down, the pressure in my abdominopelvic cavity, come on, move. Pressure in my abdominopelvic cavity, I'm pushing on it, it increases the pressure in my abdominopelvic cavity, and it comparably decreases the pressure in my thoracic cavity. And since the inferior vena cava is moving through the diaphragm, if you increase the pressure down here and you decrease the pressure up here, it's going to squirt blood up your inferior vena cava. Squirt, squirt. Every single time you inhale, you are going to squirt blood up your inferior vena cava and back to the heart. It increases venous return. And that is even more dramatic when you are inhaling really deeply and frequently, like you are when you're exercising. So exercise not only increases arterial flow, exercise increases venous return by what we call the skeletal muscle pump and the respiratory pump. And that is what we looked at right here. Okay, skeletal muscle pump and the respiratory pump increase venous return. Venous return causes stretching of those cardiac muscle sarcomeres to their optimum length. And then that, of course, is going to directly impact EDV, which is going to directly impact stroke volume. Okay, so stretching the sarcomeres, back to our Starlings Law discussion. Stretching the sarcomeres causes the force of contraction to increase because the sarcomeres get to their optimum length, and therefore you get more blood ejected. This control is not nervous system stimulated, and I want to reinforce that because we're going to see one that's similar in a minute that is nervous system stimulated. But this is just stretching them so that they are at their optimum length, and then they work better from there, and that's called Starling's Law. So basically what I'm saying is that exercise increases stroke volume, and therefore exercise increases cardiac output because it increases stroke volume. So there is a maximum amount that your cardiac output can increase above normal, and that's called your cardiac reserve. Your cardiac reserve can be increased by training, especially cardiovascular exercise. So if I remember correctly, I don't know if I'll get this number exactly right, Lance Armstrong, when he was at his prime, had a resting heart rate of, I think it was 37 beats per minute. But it actually didn't compromise his cardiac output. His resting heart rate was 37 beats per minute because his heart rate was so low because his stroke volume was so high. And one of the things that happens with cardiovascular activity is that you sort of train the efficiency of your cardiac muscle contractions. So since so much blood is leaving every time via a high stroke volume, and I think his was about 3 times the normal stroke volume at rest, then your heart rate can be really low. What that tends to do over time is you use your heart more efficiently but less frequently. It tends to prolong the health of the cardiovascular system. So athletes tend to have lower resting heart rates than people who are not athletes. And assuming there aren't any other factors involved, they tend to have healthier cardiovascular systems that will probably last them longer. Okay, so that's called the cardiac reserve. And then the last tidbit, and this is the one that everybody wants to confuse with another one, is with stroke volume. is what we call ventricular contractility. And please note, it's important that you see this, that the ventricular contractility is not related to EDV. Ventricular contractility is off over here by itself. And ventricular contractility is controlled by the nervous system. When we reveal the sympathetic nervous system over here, it'll connect to ventricular contractility. And so let's talk about this, and I want to compare and contrast it with another one. So ventricular contractility says at a given EDV, and this is really important. What I mean is we're going to be good scientists and we're not going to increase the volume of blood in the ventricles. We're going to hold the volume of blood in the ventricles constant. Even if you hold the volume of blood in the ventricles constant, there is a way to increase stroke volume that is not EDV dependent. And that is by causing the sympathetic nervous system to cause the ventricles to contract harder. And this goes back to a picture we saw before, which was this guy right here. So back to the stuff that we looked at before, which said that, okay, whereas the parasympathetic nervous system in purple here only goes to the SA node and the AV node. The sympathetic nervous system goes to the SA node, AV node, and to the wall of the ventricle. Do you see that? Get out of the way, phone. Okay, and to the wall of the ventricle right here. So what will happen is hold the volume of blood in this ventricle constant, yes? And then if the sympathetic nervous system directly stimulates the wall of the ventricle, which it can, the sympathetic nervous system causes the heart that has X amount of blood in it to contract harder when stimulated, okay? It's not increasing the amount of blood that's in the ventricle. It is simply making the amount of blood that is in the ventricle more likely to be squished out. So at a given EDV, you can increase stroke volume via the sympathetic nervous system that causes an increased strength of ventricular contraction. So now what we will see is that sympathetic nervous system does not just increase heart rate, it also increases stroke volume. So the sympathetic nervous system causes an increased strength of contraction, not just rate of contraction. Now what happens if you administer epi in a clinical situation? Epi in a clinical situation will also increase not just heart rate, but also strength of contraction. So it's really, really good to use most of the time to counteract a really dramatic drop in blood, which is called circulatory system shock. So the question, of course, like, okay, so just bringing this back to the picture that we looked at, and then we'll compare and contrast. If your stroke volume falls dangerously low, like if you have hemorrhaged or you have a weak myocardium following a heart attack, how would the body try to maintain cardiac output? So if your stroke volume falls dangerously low, you're either in shock, you have hemorrhaged, or you just had a heart attack. What will the cardiovascular system do to try to maintain cardiac output and therefore pressure and therefore flow? Well, again, what it's going to do is it's going to increase heart rate. So although it seems counterintuitive right after a heart attack when your heart is really weak, because your stroke volume fell, because you damaged a whole bunch of your cells, what's going to happen is your heart rate will go up because it's trying to maintain adequate blood flow to capillary beds. Okay, so let us compare briefly the two things that people really, really confuse all the time. And I understand why if you do confuse these things. And they are, come on, they are ventricular contractility and the concept of preload and EDV. Okay, so no, and Starlings Law. That's the two that people confuse. Okay, so one more time with these two. So please note that all of these have to do with volume. All of this stuff has to do with volume. And with Starlings Law, it says that if you stretch the ventricles, right, so that the sarcomere gets to an optimum length, then what you are going to get is a stronger contraction because of that. Okay, but this is volume dependent, yeah, and it's independent of the nervous system. Volume dependent, independent of the nervous system, okay? This one, however, holds volume constant. Do not change the volume. Just tell the sympathetic nervous system to stimulate the wall of the ventricle and you'll get more blood out without changing the amount that was in there, okay? It's just squishing it harder, independent of volume changes, okay? So people confuse these two. Don't be one of those people. Ask yourself enough questions until you think you've got it. Okay, now we'll do the easy stuff. So I think that stroke volume takes a little practice, takes a little understanding the terminology, sift through your notes. I will post those images if somebody doesn't post them first, so that you can practice with those things. There's also some pictures in your textbook that might help you. There's some flow charts in there as well. Okay, so the next thing should be actually relatively easy for you. And that is, well, how do you control heart rate? Oops. Well, you kind of know already how to control heart rate. It's not that hard because you've already learned about most of these hormones and all of that stuff. So what are the factors that are impacting heart rate? So that's where we are right here. So we just finished stroke volume, and now we're going to talk about heart rate. So what are the factors that are impacting heart rate? Primarily what impacts heart rate is how quickly your SA node and AV nodes depolarize, and what primarily impacts that is sympathetic and parasympathetic stimulation. But then there are also a couple of hormones that can impact heart rate, and they're not going to be that hard either. Okay, so let's talk about this. So factors impacting heart rate. Well, we know, and we have talked about in the last set of notes, and now we'll talk about it again, that the autonomic nervous system impacts heart rate. It impacts heart rate in ways that we can reinforce as we go through this set of notes, but it was also in the autonomic nervous system set of notes. So let's just get back to it. Okay, so factors impacting heart rate. The cardiovascular control centers are in the medulla oblongata. There is one of them, a nucleus actually, that's hooked up to the sympathetic nervous system, and there's one that's, sympathetic nervous system, and there's one that hooked up to the parasympathetic nervous system. So they're called the cardiovascular control centers. Don't let this confuse you. They're in the medulla oblongata, and they regulate both cardiac muscle contraction, and they also regulate cardiac muscle contraction and vasoconstriction and vasodilation. And they are regulating whole body blood flow. So like sort of systemic flow, not local flow, okay? So part of the medullary cardiovascular control center is generally referred to as the Cardioacceleratory Center, and the Cardioacceleratory Center just means it generally increases the heart rate. It's hooked up to the sympathetic nervous system. So it's not shown here, but this portion hooked up to the sympathetic nervous system is often called the Cardioacceleratory Center because, of course, it increases the heart rate. What it does, you already know. This one. The one that's hooked up to the sympathetic nervous system is called the Cardioacceleratory Center. What it does is it sends action potentials from the medulla oblongata down to sympathetic neurons, and then they go out and they synapse at the SA and the AV node. But again, what's special about the sympathetic nervous system and its hookup is that not only does it synapse to the SA node and the AV node, it also increases the strength of contraction because it hooks up to the wall of the heart. So that is what we were just talking about with ventricular contractility. So I'm going to make you a note right here about that causes ventricular contractility. So this that we were looking at causes ventricular contractility. So what neurotransmitter is released here and what does it do? So the sympathetic nervous system releases the same neurotransmitter here, here, and here, and goes to the SA node, goes to the AV node, and then what neurotransmitter is released? Well, you know, but let's just reinforce it. The neurotransmitter that's released right there is norepinephrine, And then what type of receptors are involved? I don't know if you guys remember this, but the type of receptors that are involved on the heart are, of course, adrenergic because they're norepinephrine receptors, but they're beta adrenergic, which will bring us to the concept of beta blockers a little bit later. Now, what is going to happen is this will increase heart rate. This will increase strength of contraction. And again, administering epi in a clinical situation will do the same thing. So administering epi in a clinical situation tends to have a pretty massive effect on cardiac output because it hits stroke volume here and it hits heart rate here. So adrenal medulla epinephrine or administered epinephrine. Okay, but then the other part of the medullary cardiovascular control center right here, hooked up to the parasympathetic nervous system, is often called the Cardioinhibitory Center because it tends to decrease heart rate. And the Cardioinhibitory Center is connected to the heart via the parasympathetic nervous system. It goes through the vagus nerve, which is cranial nerve number 10. And it sends action potentials to SA node, AV node. What neurotransmitter does the parasympathetic nervous system release at its effectors? Hopefully you know. That is our friend acetylcholine. Releases acetylcholine. And it decreases heart rate, of course. But do you see that the purple ones here? They have no direct impact on the strength of contraction because they actually don't go to the walls of the ventricle. So no direct impact on strength of contraction, whereas the sympathetic nervous system does. So they decrease the heart rate. And what type of receptors are involved in the parasympathetic nervous system controlling heart rate? Those are muscarinic cholinergic receptors. And of course, if you go back to the beginning and think about what you learned in anatomy, since generally speaking, the SA node depolarizes about 100 times per minute, but your resting heart rate is usually between 60 something and 75 beats per minute, that tells you that which branch of the autonomic nervous system is generally keeping its foot on your heart rate, and that is the parasympathetic nervous system. So if, for instance, a tiger walks in the room, the first thing I do is take my foot off the brakes, vagus nerve stops stimulating, and then my heart rate might go up to 100, and if that's not enough, I put my foot on the gas, which is the sympathetic nervous system, and I can go way above 100. Okay, so the note here is that autonomic nervous system regulation can be turned on and off by tigers, but it can also be turned on and off by other things that I'm constantly monitoring and regulating, like the blood pressure. I'm monitoring my blood pressure to figure out when to activate the sympathetic and parasympathetic nervous system, and I'm also monitoring the chemical content of my blood to figure out when to do that, and we'll cover that in just a little bit. Okay. And last thing about heart rate, and then we'll do congestive heart failure, and then this video will be over. So there are also hormones that control your heart rate, okay? So hormonal control of heart rate. The first one is easy. The epinephrine that's released from the adrenal medulla will also increase your heart rate, right? And of course, it increases ventricular contractility, and so does straight sympathetic innervation. So the adrenal medulla epinephrine, it doesn't matter how it got to the heart, whether it got there as a neurotransmitter or a hormone, it will still increase your heart rate, and that's pretty obvious. But then the one that may or may not occur to you, and it's not really dramatic anyway, is thyroid hormone also increases your heart rate. So let's look at those in your notes. Okay, so hormonal control of heart rate, epi, secreted by the adrenal medulla, which I'm fairly certain that you know by now. Epinephrine is secreted by the adrenal medulla during sympathetic activity, and it increases heart rate and strength of contraction. And then thyroid hormone right here, T3 and T4, they increase heart rate, but really just because they tend to increase the metabolic activity of all tissues, including cardiac muscle tissues. So what they tend to do is it's not like you use them as a regulatory mechanism for heart rate, but if you were hyperthyroid, you tend to have a higher resting heart rate than somebody who wasn't because of the increased metabolic activity of your cardiac muscle cells. So this one isn't like used as a regulatory hormone or anything. It just has an impact. If you're low on thyroid hormone, a lot of times your heart rate's a little lower, and if you're high on thyroid hormone, your heart rate's a little higher. Okay, and then although they're not shown on the mind map, there are other factors that influence your heart rate as well, and they are temperature, emotions, and some other things. Okay, so temperature control of heart rate. Since the depolarization business through all the way back to here, this all starts with diffusion, right? Diffusion of sodium from the extracellular fluid into the intracellular fluid. And as temperatures rise, all chemical processes, diffusion included, tend to go faster. So as body temperature goes up, your heart rate tends to go up. As your body temperature goes down, your heart rate tends to go down, just changing diffusion rates. But it's okay because as your body temperature goes down and your heart rate goes down, so do your oxygen requirements in your tissues. So that's important to understand. That's why if you had to drown and therefore cut off oxygen to your brain and your cardiac muscle tissue, it would probably be best to drown in cold water because while cutting off oxygen, you are also cutting off the need for oxygen. Not completely, but you tend to last a little longer without dying. Emotions, of course, because this part of your brain has some impact on this part of your brain, especially your hypothalamus and your amygdala. Your emotions can impact your heart rate. So it's not a freaking tiger, right? But it's a argument or it's a stressful job interview. Your emotion can directly impact your heart rate via the autonomic nervous system, and then also via stress hormones that are released, both epi and then eventually cortisol. Okay. And then the other thing is also true. This only works, this whole story that we've been telling relating to ions and heart rate, if the ions that are involved in controlling your heart rate are normal. If your potassium is off, it will have an impact. If your sodium is off, it will have an impact. If your calcium is off, it will have an impact. And then, like we said when we were talking about Lance Armstrong, if you are training for endurance over long periods of time, you can actually increase stroke volume, which allows for a decrease of heart rate. Okay? And then last thing today, and then I'll finish the other part next time, is I want to talk just a little bit about congestive heart failure. So congestive heart failure is really interesting. Okay, so let's see if I can explain this. I usually flap around in the room to try to explain it, but I don't have much flapping room here. All right, so what happens with congestive heart failure is that the ventricles generally are going to weaken. Let's say that is due to a heart attack or due to too much pressure in the arteries that you're dealing with here. When the ventricles weaken, what happens is the blood moving out can't get out as effectively as it does moving in. Because please remember that venous return has mostly to do with skeletal muscle contraction. Passive venous return through the veins is less dependent on what the heart is doing than arterial flow out is. Okay, so the venous return back to the heart, skeletal muscle contraction, you're always breathing, so the blood's always coming back in. So the blood continues to come back in and it continues to come back in, but then it can't get out. So you have congestion, right? So it's like a traffic jam, and the traffic jam is really in the ventricles. And so what happens is blood starts to back up, and it backs up, and then it will eventually start to leak out into the capillary beds that we're feeding back into the heart. So I'm going to show you guys a little animation of congestive heart failure. Where is it? There it is. Okay. I think this is made for patients, so it's not super detailed, but I can use it to explain this to you. So congestive heart failure. Ready?
That pumps oxygen and nutrient-rich blood to your body.
Hold on.
Your heart is a beating muscle that pumps oxygen and nutrient-rich blood to your body through the circulatory system. Inside your heart are 4 chambers that are responsible for collecting blood and then redistributing it to your lungs and body. Several conditions can impair the heart's ability to effectively circulate blood. Defects or diseases of heart muscle or the heart valves and coronary artery disease can result in a failure to maintain an adequate circulation of blood to satisfy the needs of the body. And high blood pressure and narrowed or clogged arteries can increase the resistance to blood flow through the vessels, thus increasing the workload of the heart. Any of these conditions can result in an enlarged heart with compromised function. Eventually, the failing heart cannot pump out what is being delivered to it, and pressure builds within the heart chambers and venous system, causing an accumulation of fluid in body tissues. This progressively worsening series of events is called congestive heart failure. As CHF progresses, swelling often occurs in the legs and feet, and fluid may collect in the lungs, causing shortness of breath. Eventually, heart failure may affect the function of the kidney, further increasing fluid accumulation and perpetuating this unhealthy cycle. The symptoms of CHF are treated using diuretics to eliminate excess fluid. The primary cause of CHF will then be targeted using medications to increase the pumping ability of the heart or decrease blood pressure, thus lessening the amount of work the heart has to perform. In the case of valvular defect, surgical intervention may be required.
Okay, so that's just giving you guys a visual of what happens with CHF, and then I'll show you a little bit about it here too. Okay, so CHF can be caused with problems with the heart. It is also often caused with the development of atherosclerosis and arteriosclerosis over time. And so as the pressure gets really high in the vessels, your heart has to work really hard to overcome that pressure. Pressure getting high in the vessels usually has to do with arteriosclerosis because of a buildup of cholesterol and cholesterol deposits. And so what happens with CHF, and you'll probably be dealing with patients with CHF at some point in your career, is the backup into the capillaries of the lungs causes fluid to the leaks in the lungs, which causes pulmonary edema, shortness of breath. It's really hard on the heart. The heart will increase in size. You'll also get fluid that is pushed out of systemic capillaries into the hands and the feet, and you can also get some swelling in the abdomen. And then later on, your heart has been working so hard to try to compensate for this traffic jam that you'll get an increase in the musculature and the walls of the heart. So this is a transverse section through, this is the right ventricle and this is the left ventricle. And so this person had congestive heart failure of the left heart, and the heart was trying to work really, really hard to compensate for that. And you get an increase in the musculature of the heart, but it doesn't really help, because what it eventually does is it actually reduces the size of the lumen of the ventricle, so you can't get as much out. So last little tidbits about this is that if you ever have a pressure problem, you will see that basically the ways of treating a pressure problem are to treat the heart. This is a medication right here called Digitalis, which slows down the heart for more filling time and therefore greater stroke volume. You can also treat the vessels, give vasodilators to decrease the pressure that the heart is pushing against, or you can treat the fluid volume. So when we're dealing with pressure, and we'll see this in the next video as well, there's basically 3 ways to manipulate pressure, either with medical intervention or internally. Mess with pressure with the heart, the vessels, and the fluid. Okay, so the last thing that they can do, and often do, with CHF is give diuretics to reduce the amount of fluid in the system. So this slows heart rate, this causes vasodilation, and this actually reduces fluid volume. Okay, so last tidbit is failure of the left heart. If the left heart isn't working, what you have to do to figure out where that would cause problems and leakage is to back up from the left side of the heart to the capillaries that fed into it, which would be in the lungs. So this causes pulmonary congestion or edema and shortness of breath, especially when you're lying down because of the fluid that's backed up into the capillaries of your lungs. And then failure of the right heart causes what's called peripheral congestion. It backs up into your systemic capillary beds, but where you often see it is the hands and the feet. It's hard to get that blood back anyway. So swollen hands, swollen feet, even in the morning, and even when you did need a bag of Doritos the day before. Okay, so next time we will finish and we will talk about extrinsic control of blood pressure, nervous and endocrine control. I'll do that for you guys next time. And then I will tell you which day your test is going to be. I'm probably going to give you like 2 days to do it, but once you start it, you have to finish it. Okay, I will see you in the near future.